Yeah, so I guess I don't think anyone else is coming for a while yet, so we'll see. All right. All right, so let's get started. Yeah, a couple things to talk about. I actually run across the new term for Susan, Vertex biology, which is where you have the trijunctions and the junctions between cells. So we'll cover some papers on that.
Oh, wow.
Yeah, I don't know. Yeah, who coined that term, but it's a term now.
Oh, really?
Yeah.
Huh.
All right. All right. So the first thing I wanted to talk about was this article on
Horse.
that I did for Darwin Day, a review of carcinization from the biology to computational models. And so what carcinization is it's basically the idea that you get a lot of phenotypes that sort of default to crab phenotypes. So this is an image of different marine invertebrates, and the idea is that this crab phenotype is convergent. It exhibits convergent evolution over a number of different clades. So you see the crab things kind of default to the crab phenotype, and that's kind of this phenomenon that they talk about in the literature. And what I did is I reviewed not only that phenomenon in the biology literature, for which people have worked out the phenotype, kind of defining what carcinization is, but also talking about the genetics of carcinization and some of the genetic mechanisms that are involved. And then what's interesting is they also have computational models of carcinization. So they have these types of network models that were particularly interesting. This is a picture of these developmental network topologies um, from egg to crab phenotype. So what they've done here is they've started with the egg, which in marine invertebrate is this egg not pleus, and then there's the zoea, and then there's this megalopa, and then there's this crab phenotype. So you have these transitional phenotypes from the egg through development, and you see that this is based on the phenotype, and you can see that there are these different topologies that emerge over different life stages. We also have, this is the part about gene regulatory networks, and then that leads into a discussion on epigenetic landscapes and how we can use epigenetic landscapes to understand the evolution of these phenotypes, which leads to a few other computational models in the literature where people are looking at this genotype to phenotype mapping and how that genotype to phenotype mapping is convergent between many different genotypes to the same phenotype. And so this is an interesting article because I actually got through a lot of different references here, both computational references, biological references, and genomic references. Just a blog post, but yeah, it's next thing I wanted to point out was that we have our DevoLearn repository. So for those of you interested in Google Summer of Code, we have our DevoLearn repository. We have, I think we talked about this last time. We have the DevoLearn program, which is something that we worked on in Google Summer of Code from 2020 to I think 2 years ago. So we've been kind of moving back and forth between DevoGraph and DevoLearn, and sometimes people have had to add to DevoLearn to get their things in DevoGraph to work. So DevoLearn is a series of pre-trained models, a series of deep learning models that allow people to segment microscopy images. So if you have a microscopy image data set that you're interested in segmenting, we can segment this with the DevoLearn program and include it in the same pipeline as the DevoGraph program. And so this is sort of what was at one time called the stage 1 of DevoGraph. So if you see references to stage 1 in the documentation, that's what this refers to. So this is something that's been released on PyP. We have a number of different models here to work with. Those are also on Hugging Face. So we have we have a bunch of different. So we have our links to the Hugging Face spaces. We have the segment the cell membrane in C. elegans embryo. Oh, I'm sorry. So we have the links to the Hugging Face spaces. We have segmenting the cell membrane in C. elegans embryo. We have the cell membrane segmenter for that. Segmenting the nucleus in C. elegans embryo. We have the C. elegans nucleus segmenter for that. And then the cell lineage population prediction. We have the lineage population model for that. So we have a segmentor, a membrane segmentor, a nucleus segmentor, and a lineage population model. And so those three models will allow you to extract data from an image file and produce an outcome. And so if you want to contribute to this, that's open, open to contributions. And you may need to do that to do things with DevoGraph. Now, the state of the art on this, of course, is the segment anything model. So Meta's segment anything model, it's been out for about 2 years. We've kind of I think the way this worked is we forked a biological version of the segment anything model, and we've kind of refined it for our own needs. So what we're doing in a lot of the things that we've tested it with and benchmarked it with, we've done things with the C. elegans embryo. Now we'd like to move to other embryos, but the problem is that these are sometimes very different modes of development. So if we train or pre-train a model on the C. elegans embryo, we'll get certain results, and we have an annotation of each cell. But if we move to, say, like a Drosophila embryo or some other embryo where we don't know the cell lineage, or it's not an exact cell lineage as we have in C. elegans, then we run into some problems of application and accuracy. So, we've been kind of focused on C. elegans. If people are interested in contributing to this package in a way so that we can do other embryos, other types of developmental models, you're welcome to do that. But that's not really the focus of the GSOC project. So that's DevoLearn. And you can dive into that if you'd like. We have a repository for CellSam, which is the biological instance of the segment anything model. This is our fork here. And we just, if you want to just use CellSam, that's fine too. We can, I think that's kind of sort of maybe less C. elegans focused than, it's a more general model than DevoLearn at this point. But, this is where we've kind of drawn from this fork of CellSam. And then, of course, we have our different models here, nucleus segmentor, membrane segmentor, and lineage models. Those are also on our GitHub, but also on Hugging Face as well. So this is the DevoGraph repository. This is the focus of this year's Google Summer of Code. We have a number of different directories here. We have the two projects, Growing GNN and Hyper DevoGraph. Growing GNN is the one where we've forked the neural developmental programs repository, and we've also forked some other repositories on topological data analysis. And we're building these embryos that grow in terms of the cells and the cells that are visualized. So this is what your growing graph looks like. You have the nodes, you have the edges, the vertices and the edges. They're labeled according to sort of the C. elegans lineage tree. And as the network grows in size, it gets more dense. And you can see that it's plotted in this three-dimensional space that shifts over time. So that's what we're trying to produce. We're trying to produce this model of the embryo as the cells divide and this network grows in size. And we can play with this network so that we only have the cells from one stage of development, or we can have these graphs. Usually the way that we plot them here is where all the cells subsequent, or all the cells prior to the current stage are plotted. And that's just to show where those centroids were in the embryo space. So that's what this is. So this is something that we've been working on. This repository, Growing GNN, should give you some detail. This is Paki Banchalia, who was the person who did this last year. And so we have some nice documentation for this. So we're basically combining temporal growth with spatial learning, and that's what we're doing here. And this is again applied to C. elegans data, so we can look at this for other embryos, we can look at this for other systems, but for right now it's kind of specialized to C. elegans. So there are some details to work out with respect to C. elegans data. I think Paki only got to maybe about the 128 cell stage. They're actually 660 some cells at hatch. So the worm grows, the embryo grows until the egg hatches, and you have many more cells by the time the embryo hatches than you do at the stage at which we have a representation here. So we need to work on that latter part of development, which is harder because it has more cells, and that may not be possible because we don't have access to really high powered computing. If people, one possibility is to improve the code so that it'll work for larger data sets more cheaply. That's a possibility. We have a fork of the original repository. Another possibility is to try to visualize some of the things that we see in early development. So there are a lot of possibilities with the growing GNNs part of this. Then we have the HyperDevo graph, which is this where we're using hypergraphs to model C. elegans development. This is Mehul Arora, and I know a number of people have already expressed interest in this. This is where we build these hypergraphs, which are graphs within graphs. So you have hypernodes, which contain regular nodes. You have hyperedges, which contain regular edges. And you can build these multi-scale representations. So this is something that Mehul has worked out the details on. And we've produced some nice visualizations. So these visualizations kind of look like this, where you have, we have the cell nomenclature, we have the cells represented by a hypernode, and then there's this radius by which, we include all the cells in that radius. It's kind of hard to see the graph part of this, but these local overlaps and these concentric circles are basically the edges and the hyperedges. So we need to work on the visualizations. We need to work on how, Mehul has done a lot of work on creating metrics, graph metrics, and other types of metrics, trying to get into some benchmarking and then the visualizations. But I think the work that can be done, of course, is making working with the code, making it more efficient. The visualizations, if we can work on those some more, and also kind of benchmarking this against other. I think Mehul mentioned that we need some better benchmarks. And so we can work on that. That's something you can propose. Finally, the thing I'd like to talk about with respect to DevoGraph is that we have, if you explore this repository, you'll see that there are these different stages. So you have stage 1, stage 2, and stage 3. So this is kind of old, but it's still relevant. Stage 1 is kind of already been done, but you'll find a lot of things here with reference to image processing and a lot of connections with DevoLearn. So that's why I talked about DevoLearn. So basically the tools of DevoLearn, the tools of Segment Anything are stage 1. Stage 2 is this visualization step, creating the networks. This is something that we did with topological data analysis. There's a tool, KMapper, which has been used in years past that we can use as a visualization tool. And then stage 3 is kind of moving towards hypergraphs. And there's a TDA folder here. I haven't updated this to reflect kind of how HyperDevoGraph and the growing graph neural networks fits into stage 3. But that's basically it. It's like that last stage, that most complex stage of the project. So yeah, this is basically applications of graph neural networks to developmental biology. And so there are a lot of ways people can contribute to that. If we go to our Slack channel, I notice that Morgan Hough has contributed this YouTube video, Hypergraphs Rewriting for Well-Defined Geometries. So this is very much in the spirit of what we're doing in DevoGraph. Take a look at that. We also have some readings and some videos on graph neural networks in the OpenWorm Slack channel. So if you join the OpenWorm Foundation Slack, and I know a lot of people who are coming through NeuroStars for Google Summer of Code, they're encountering this link to the OpenWorm Slack. And so if you join that, if you click on that and get an invitation, you will get access to this Slack. And it has a lot of, of course, our meetings and then some of these other things. I wanted to point out that Hussein Ather has forked 2 of the repositories in our DevoWorm GitHub. So we have a DevoWorm GitHub that's separate from the DevoLearn Github. And the DevoWorm GitHub is just kind of legacy projects, legacy repositories. He forked 2, the Digital Basilaria project, which we haven't talked about in a while. And that is a trying to digitally represent a genus of diatom called Basilaria, which is this cool colonial organism that moves in a very unique way. So if you're interested in that, that's not part of this year's Google Summer of Code, but I am glad to see that he forked that. So if you're interested in that, let us know. It's just kind of one of those projects that hasn't been touched in a while. And then we have this other repository, Theoretical Types of Embryo Developmental Networks. That's kind of linked to the DevoGraph stuff. It's a link to some of the stuff I showed in the blog post on embryo networks, where you take the centroids of cells in an embryo and you look at how they're connected by some criterion, either by distance or by some signaling criterion or something else. So congratulations to Hussein and Morgan and anyone who's applying to Google Summer of Code this year. So I think the descriptions are now live on NeuroStars. So if you go to neurostars.org and you go to the GSOC topic, you'll find these projects or the Graph Neural Networks project listed. And so go through that, leave a comment if you'd like, or attend one of the meetings live, or raise a comment in the Slack. It should be pretty good. We should have a pretty fun summer, at least, in terms of thinking about these problems.
Don't have a question. I got my diploma in the mail and I've graduated.
Oh, congratulations.
Yeah, I don't know.
Well, it's good. Yeah.
Yeah. I don't know. I'm still working on things.
Yeah.
I have my thesis presentation is going to be presented in APS March meeting on the 19th in the evening. And they put me in the evening section. I don't know if they knew what to do with me. But anyways, it's a 10-minute presentation.
Okay. Well, good luck with that. Is that just kind of a summary of your thesis then? You're going to like condense that?
Yeah.
Great.
So that, and doing that, I figure a master's thesis, one should write a paper about it and one should present it. So that's my presentation. And then I'm writing a paper.
Oh, good. Yeah. That's great. So you're working on the paper now? Or?
Yeah. The direction Dr. Zhang wants to go with it and the direction I want to go with it are two kind of diverging problems. So he thinks he wanted to, well, I thought maybe we can make a summary of what type of morphology cells have, like how they're connected together, but it's patterned and it's different for every tissue, and that gets into another thesis perhaps.
Yeah.
So I'm not sure I can do that part of it, but maybe I should just do a summary of my thesis and that's it.
Yeah. Well, yeah, there are a lot of kind of potential questions there. And of course, like applying the tensegrity method, people are going to ask you about different questions and then you apply it to different questions. And there's that risk of kind of using it as a hammer for every nail. But I think you could probably find a good set of things to work on. Yeah.
No, you can apply it where it shouldn't be.
Well, yeah.
Yeah. You can take my model and make it, well, what I call its K-factor, so extreme that maybe it wouldn't ever exist in a cell, and then apply it to, well, I did apply it to skin. It kind of matches skin, but really what it needs is a larger model. Like instead of in 1 micrometer, 2 micrometers, or 3 micrometers, it needs to be like whatever skin is. Like how thick is that?
Yeah.
Like 20 micrometers. It needs to be a larger model.
Yeah.
It's still extreme because it's the contrast between the bars and strings in the tensegrity model.
Right.
So the elasticity has to be very different between the bars and the strings in the model for it to have a different slope and area where it works. And some of that just a little extreme. I don't think it actually exists. So you can make a model that doesn't actually exist in real life and apply it to tissue. I don't know. Wasn't the idea. I wanted to make a kind of a real life model, like something that has actual foundation in reality.
That's what we hope for, yeah.
Yeah.
Well, yeah, that's good. And, I was kind of wondering about the computational power you might need for a larger model. So is that something you think is tractable, or do you think it's something that, like, you would have to struggle with?
It's possible. Like, my model solves in seconds. So you could probably you can make it a lot larger and it would still work. Should be able to, except it's a tensegrity, so it all has to fit together and work.
Yeah.
And finite element analysis tends to not work with tensegrities because it's not stable enough. You'd have to change the programming for the finite element program code. You'd have to change the code.
Yeah.
So I don't know how to do that.
Well, yeah, it seems like a whole ordeal.
Yeah, I haven't looked at it. I went to a conference on tensegrity, and I have a lot of people's work with it, and they were programming in Mathematica for one thing, and in MATLAB, and Python. So I haven't looked at that yet.
Oh, well, yeah. Good luck. Yeah. I haven't seen any I haven't seen any Python models for tensegrity. It doesn't mean that someone hasn't tried it.
Well, they tried a bunch of things. They were did code in another finite element analysis program. It wasn't ANSYS, it was something else, but people have programmed in something called ANSYS, with finite element analysis. COMSOL's finite element analysis, and so's ANSYS, and so's this other program they were using.
Dick said, search free supercomputer access.
Yeah, well, if I made the model 1,000 cells large, then maybe I would need one. But it's not with 5 or 7 or 14 cells. It's not gonna. It'll just solve on a regular computer.
Yeah, the way we usually do it here is like with interactive notebooks, like different types of collab notebooks, and you get like computational power like for free from the cloud server. I mean, there's a limit to what they offer for free, and you have to start paying. But that's usually though those kind of notebooks compile Python and maybe some other languages, but specialized languages maybe not. So that doesn't help. That's why I was asking about Python, because that's more of a standard where you can get like toolboxes and things like that.
Yeah, it's really easy to convert from MATLAB to Python. They're very similar.
Yeah.
Very similar codes. Same structure.
Yeah.
I don't know if I want to get into it or not.
Yeah, it's similar, but not.
Yeah, you still have to convert it.
Yeah.
Yeah. Time consuming.
Well, good luck, yeah.
Yeah, I have a Udemy course on neural networks, and he's actually studying the brain and the signals from the brain. And his code, he does it both in Python and MATLAB. And they're just very similar.
Yeah, they are. Yeah, it's but definitely it's hard to get them to sometimes it's hard to convert things because Python is a more formal structure, or especially if you get deep into a program.
Okay. Well, I don't know Python. I've learned too many programming languages in my life, so I'm resisting.
Yeah.
After you know 10, why do I want to know another one? But I guess Python might be worthwhile.
Yeah. Yeah.
I don't do anything with my computer programming.
Yeah.
I'm good at it according to my marks and what I do in class, and then I go out in the real world and they don't want me to use it for anything. So, it's useless. It's just a hobby, just like a few other things. Computer programming is a hobby.
Yeah. Dick said you could use AI to convert it, but I don't know. Well, sometimes
Well,
people use.
yeah, apparently there is some AI to convert that, but it's so similar, at least in the code I've seen, that you can just go, oh yeah, and just change that.
Yeah. Yeah. Okay. Well, thanks. Yeah. For the update.
I don't know.
Yeah. All right. Let's move on to the vertex biology I was talking about. Okay. So there are a couple of papers here I'm going to cover. We have 4 of them here. Let's see. So we'll start here. Um, this is a paper by Sugimara and Otani, vertex remodeling during epithelial morphogenesis. So this is kind of where they talk about vertex remodeling. So this is where the vertices are moving and changing their topology with respect to an epithelial sheet. So we'll go through the abstract and then maybe down through some of the details here. So the abstract is here. Epithelial cells adhere to each other by intercellular junctions that can be classified into bicellular junctions and tricellular contacts or vertices. So you get these bicellular junctions, 2 cells. You get these tricellular contacts, which end up being vertices in a graph. And so they talk about this actually down in this part of the paper here. So we'll get to that next. Epithelial morphogenesis involves cell rearrangement and requires remodeling of bicellular junctions and vertices. So morphogenesis in the epithelial sheet, sort of the cells move around, they change their shape, they change their contact, which is important, and then they remodel these junctions and vertices as they do so. Although our understanding of how bicellular junction mechanics drive epithelial morphogenesis has advanced, so our understanding is we're getting to the point where we're giving it a name, vertex biology. The mechanisms underlying vertex remodeling during this process have only received attention recently. In this review, we outline recent progress in our understanding of how cells reorganize cell adhesion and the cytoskeleton to trigger the displacement and resolution of these vertices. So there's cell adhesion, there's reorganization of the cytoskeleton, as we know, and then that triggers this displacement and resolution of these vertices in the sheet. We will also discuss how cells achieve the optimal balance between the structural flexibility and stability of their vertices. So there's this trade-off between being able to have these vertices that reconfigure and then having vertices that are stable. So if you have a tensegrity structure, then we can maintain that tensegrity structure and it doesn't move away from that configuration. Finally, we introduce new modeling frameworks designed to analyze mechanics at cell vertices. So they're actually building a formal model. Integration of Lyme imaging and modeling techniques is providing new insight into the active roles of cell vertices during epithelial morphogenesis. So this part here, this paragraph is interesting right here. Cell-cell junctions between epithelial cells are classified into bicellular junctions where 2 cells adhere to each other. So this is where we have this adhesion principle. And then tricellular junctions or TCJs and multicellular rosettes where 3 or more cells meet. So these are these junctions that Susan's talked about where you have multiple cells meeting at one point. The term TCJs has historically been used to refer to tricellular occluding junctions. So this is where you have trijunctions, but they occlude one another, where the cells are all kind of working against one another. They're occluding one another. So that's kind of a different arrangement. However, recent discoveries of the tricellular adherens junctions, or TAJs, in invertebrates raise ambiguity in the term of TCJs. So we have these occluding junctions, which are a broader class of these tricellular junctions. We also have these adherence junctions that exist in invertebrates. And so there's this sort of ambiguity in the term. And so I think, as usual across biology, complexity is intervening in our classification systems. And, people have to kind of rethink things a bit to get a better handle on what we're dealing with. In this review, we refer to tricellular contacts in general as vertices and junctional specializations at vertices as TCJs. So this is their definition: tricellular contacts as vertices, and then junctional specializations at vertices. When distinct junctional structures can be recognized by electron microscopy, or when the structure contains unique molecular components, the vertices are referred to as tricellular tight junctions. So I think Susan also talked about tight junctions and sort of the role of that in epithelial sheets, tricellular separate junctions, and depending on the ultrastructural feature and the molecular composition, you can have either one or both. Actually, it's tricellular septate junctions. Tricellular contacts are also observed in mesenchymal cells, although they likely lack distinct junctional specializations at ultrastructural levels.
I ran across an interesting note along the way somewhere where they said that there are adherence junctions in between. There's adherent junctions and tight junctions, and they're kind of mixed together. And when the cells are hatching, and like mammalian cells are hatching out of the zona pellucida, that the adherence junctions don't exist, like they dissolve.
Okay.
So that's an interesting thing, because that's part of how cancer metastasizes as well. Like they don't go through a small opening. And I wanted to tell Dick this, but he's occupied. Anyway, so yeah, I went and lost the reference, but the adherent junctions dissolve so the cells can go through, or the tissue can move through wall space.
Yeah.
So the trijunctions are important. Anyway, yeah.
Yeah. All right. Yeah. Thanks. So the rest of the paper is this is a figure here. This shows the structure of a tricellular junction, where you have some tissue deformation. This is supposed to be a tissue sheet A. You have the cell shape change, where you get these junctions. You see the white dots, and those are the junctions. Here, the other cells are converging on this red cell. The white dots are these junctions, so where they meet together, the edges of the cells. So if you think about it like, like a tiling in mathematics, where you have cells that are kind of tiled together, and you have to see how they fit together without huge gaps in between. That's what these are, and then the junctions are just the points at the vertices of the network. And so the cell shape can change, of course. It can be squeezed in different ways, and those junction arrangements can kind of move relative to one another. And then these cell arrangements are, of course, important in multicellular organisms, where you have these different stable configurations. So here we have our cells represented as hexagons, and then the junctions here, and then this is where the cell kind of moved. These two cells on the vertical axis kind of move together. They form a different junction, and then there's this rotation of this sheet here, and that forms a different orientation of junctions. So
there are also 5, 7 instead of hexagons.
Yeah.
If they're eight-sided, there's also 5, 7 configurations, and they're more fluid.
Okay.
Anyway, They still have trijunctions.
Yeah. All right. Then we have two examples here, invertebrates and invertebrates. So they mentioned that invertebrates, you have these different specialized types of adherence junctions. Right? So you have adherence, and then what was the other one? The SJ. So we have the AJ and the SJ. And so we also have the TJ, so the tight junctions, the adherent junctions, and then the SJ. And so these are all kind of specialized in these sheets. So in invertebrates, the mature epithelium has these different layers of adherence junctions, and then the SJ, and then we have invertebrates, we have the trijunctions here, and then the adherence junctions below. So we're looking from apical to basal in the sheet. And you see that there are these different types of configurations as one moves down the sheet, and invertebrates and invertebrates. And this is mature epithelium, so that arrangement is very different in invertebrates versus vertebrates. Okay, so that's basically the idea. And then this just shows like a trijunction, and this is the actual junction that you see here in this micrograph. It's like right here. And then that's this cartoon where you see the actin bundle kind of along the edge of the junction. So it follows the boundary between the cells and then kind of skirts the junction in different ways. So there are these actin bundles that attach to the wall, to the membrane, and to the intersection of the cells, and then you have the tight junction here, or the trijunction here. And so you can see that actin plays a role in kind of configuring these junctions. Did you have anything on that, Susan?
I tried to make a model like that with the actin bundles around the corner like that, and it fell down or something.
Oh, okay.
Yeah, it didn't work. But then a standard tensegrity, it's kind of in a star shape, also never works
Oh.
in COMSOL. Here we go again. Well, yeah, I have trouble with my video. Anyway, so again, finite element analysis does not work well with tensegrities.
Yeah.
And it needs a different boundary conditions or something, because it doesn't fall down. It is merely changing configuration, because you put little finite forces on it until one of the strings becomes loose for whatever reason in the configuration, and then it will all of a sudden change shape, and it has not fallen down and broken. It is merely changed shape like tensegrities do. And so that has to be programmed into your finite element analysis, or is going to be very difficult to do tensegrity in finite element analysis. Yeah, so I've nearly convinced myself that I should do some work in Morpho, which is out of Tufts or Boston, where they've got a program, but it's I don't know, it's that line code program. Okay, I'm forgetting what it is.
Okay, yeah, let's.
A programming language that I don't know.
Yeah. All right, so there are a couple other papers here. That's that paper. I'll send all these papers out after the meeting. This is another paper, tricellular junctions, how to build junctions at the trickiest points of epitheliosis. TRIC is TRIC, and that's a kind of an acronym, so they're being cute here. And so this is, I guess, their model. And then this is Higashi and Miller, I think at University of Michigan. So in this case, what they're looking at is they're looking at these tricellular contacts. These are places where 3 cells meet, like we just saw in the last paper. Invertebrate epithelial cells, and we talked about that there's a difference between vertebrate and invertebrate epithelial cells. Specialized structures called tricellular tight junctions, or TTJs. We saw that graph where we saw the role of tight junctions at the sort of the apical part of the sheet. And tricellular adherens junctions, which are lower down, TAJs, have been identified. TTJs are important for the maintenance of barrier function, and disruption of TTJ proteins contributes to familial deafness. This is in a phenotype. So I guess this is in the ear where they're looking at this. TAJs have recently been attracting the attention of mechanobiologists because these sites are hotspots for epithelial tension. So this is where you have tension in the epithelial sheet, and that's kind of the place where that originates. And in the, I guess in the inner ear, that would be pretty disruptive if you had, differences in epithelial tension. Although the molecular components, regulation, and function of TTJs and TAJs, as well as of invertebrate tricellular junctions, so they're talking about vertebrate structures in this first example, and then the invertebrate tricellular junctions are also being characterized, as we saw in the figure. Many questions remain. Here we broadly cover what is known about tricellular junctions, propose a new model for tension transmission at adherens junctions, and discuss key open questions.
That's the way I made my model, with the trijunctions connecting the cells together, bearing the mechanical load.
Yeah.
Maybe that's why I got interesting results. See, that's in my thesis. That was diagram here, B. And put that in my thesis. Yeah, I put that in my thesis.
Okay, good. So this is A. This is a picture of kind of these junctions using angulin 1 as the probe, as the stain. We have ZO1, which shows the membranes. So we have the dots in represented by angulin 1. ZO1 represents the edges of the membrane. So you can see that those dots are just kind of the intersections of these membrane edges. And then merging that, you see the junctions in relation to the edges of the membranes. And then this is Susan's favorite, where we have this is in 3D, so we have these cells and the tops of the cells, but also the depth of the cells to see these junctions, kind of how they sit at the, they sit. If you see this image here, it kind of sits at the top of the sheet, but there's some depth here as well. So the junctions affect the depth along these axes. So this is the apical to basal axis, basically where these junctions are acting. So the cells can twist and bend underneath, of course, and we can have these occlusions where cells will kind of move over another. And so you have to think about that when you think about this relationship. And then we have the tricellular junction components. We have tricellularin, which is a protein, angulin 1, 2, and 3, and those are all different proteins. We then have this section of the three-dimensional tricellular junction blown up. We have invertebrates. So in vertebrates and invertebrates, of course, we have different structural components where we have TJ, AJ, which are the adherens junctions, the tricellular junctions closer to the top there, and then showing that in vertebrates, where we have ECAD complexes and these TJ strands, and these are like the actin that we saw in the last paper. And then this is SJ strands here, and ECAD complexes on top in the invertebrates. So they don't have actin in the same way that vertebrates do. And it's just interesting how that they have the same junctions, but the molecular component underneath is different. And then this figure F and G are where we have a model of tricellular junction organization in F. So this just shows the anguin and the tricellularin as components of this geometry. And then in G, we have model of TSJ organization, where we have anaconda and gliotactin as different molecules in this organization. So you can see that these different types of models kind of propose different ways in which the structural integrity is maintained of these junctions. Okay. And then this figure is where they kind of look at how tricellular junctions are formed. So they're formed kind of from cell divisions. In A, you see a cell division where you get the single cell with tricellular junctions. The cell divides, and you get these different structures that occur at the boundary of the membrane, and then you get these new junctions that form. So the cells are always migrating, but they're always also always dividing, or they're dividing in some cases. And so you get these different opportunities for new junctions to form, for old junctions to sort of rearrange themselves. And so and then we have an example from convergent extension here, where a sheet of epithelial cells stretches out along one axis. And so convergent extension, you can see that like these different colored cells that are kind of arranged in a parallel fashion then sort of interdigitate here during convergent extension. And they form these trijunctions in different places. So you can see that's, this is a component of morphogenesis where you get this rearrangement of the epithelial sheet, and in a way that kind of reconfigures these tricellular junctions, and in a way that kind of changes the function of the tissue eventually. Okay, so that's that paper. Next is this paper here. This is a figure. This is the one that we just saw in the last paper. And then this paper here, which is the last example. This is called, this is from the archive. Trajectory geometry suggests cell fate decisions involve branches rather than bifurcations. So I'll just do the abstract on this, and then we can talk about this, and that'll be I'll send these papers out, and we can talk about them maybe next week as well. So trajectory geometry is the name of their program. I don't want you to think that that's a typo, but the abstract reads, differentiation of multi-potential progenitor cells is a key process in the development of any multicellular organism and often continues throughout its life. It is often assumed that a bipotential progenitor develops along a relatively straight trajectory until it reaches a decision point where the trajectory bifurcates. So we have these cells that are differentiating, and they can differentiate into different types of cell. We have these progenitor cells, and then we have these cells at different fates. So a progenitor cell can differentiate into different fates, and it can, this is something that we want to model alongside this sort of, these sheets, these cellular sheets and their configuration. It is often assumed that a bipotential progenitor develops along this relatively straight trajectory. At this point, one of two directions is chosen, each direction representing the unfolding of a new transcriptomic program. So we have these transcriptomic programs that turn on as these different fates are selected, these different intermediate fates are selected. However, we lack the quantitative means for testing this model. Accordingly, we have developed the R-package trajectory geometry. Applying this to published data, we find several examples where, rather than bifurcate, developmental pathways branch. So this is where I don't know what the difference is necessarily between bifurcation and branch. That is, the bipotential progenitor develops along a relatively straight trajectory, leading to one of the potential fates. A second relatively straight trajectory branches off from this towards the other potential fate. In this sense, only cells that branch off to follow the second trajectory make a quote-unquote decision. Our methods give precise descriptions of the genes and cellular pathways involved in these trajectories. We speculate that branching may be the more common behavior and may have advantages from a control theoretic viewpoint. So this is a little bit different than talking about the sheet itself. This is talking about how cells go, from a progenitor cell to a differentiated state, and the multiple differentiated states that a lot of cells will undergo, especially in mammals. And so they're talking about these lineage decisions and the enteric nervous system. And this is, in this system, we've uncovered a novel configuration of differentiation trajectories. So this is where we have this bifurcation model, which is where a cell develops towards the chosen fate once a fate is chosen. So a typical model of cell fate decisions is as follows. A multi-potential progenitor cell develops until it has reached a decision point. Here the cell chooses one of two possible fates, each of which requires the initiation of a new developmental program. So this is the bifurcation model. Then this other model here is this where you have these trajectories. In contrast to the bifurcation model, enteric glycogenesis forms a default linear pathway of progenitor maturation from which neurogenic trajectories branch off during embryogenesis. A consequence of this branching configuration is that there are no identifiable points of commitment along the gliogenic trajectory, and it is only the cells which become neurons that ever make a decision and initiate a new developmental program. So rather than from a single branch point, there seems to be a region along the default trajectory where branching can take place. So this is kind of an example of this in figure 1, where they use synthetic data to show these different bifurcation and branching models. So this is where we have in A, a simplified model of branching behavior, which is where we have a cell that makes a decision to go to differentiate into one of two cell fates. B is a simplified version of branching behavior, is a model of cell fate decision. And then C, this is where bipotential cells proceed along a default pathway to one of the potential outcomes. So this is where they don't make a decision, they just kind of move either to the blue or to the green. So in this A, there's a decision being made at this branch point. In B, you just have this deterministic process, I guess, where they either follow one pathway or another. And then in C, we have branching behavior, where we have this region rather than a single point, in which cells choose to leave their default pathway. So you have these 4 points here, where you have, decisions that are made between the blue and the green. Okay, so it looks like we have new people here, so welcome. So any questions about those papers? Susan just posted a reference.
It's an older reference, but it was interesting because the joining of the 3 cells or 2 cells or whatever is usually depicted at the apex of the cell, and this one model shows, or the paper here was showing that on a stiff surface, the join between the cells is actually at the basal surface, or near the basal surface instead. So I thought that was interesting. It's also something I could try with my model.
Yeah, that would be good.
Yeah.
So I think there's a lot of potential in looking at the geometry, looking at where different places in the 3-dimensional geometry where those things happen. So it's probably a huge difference, I'd imagine.
Well, I'd like to know more biology, more papers on where these things are and their strength, and for instance, the strength of the actin bundles, like what is its elasticity, and the literature isn't there.
Yeah,
Like
you can see that
a
like
turtle.
they have to use kind of a lot of diagrams and a lot of synthetic data to get the parameters aren't really available. That's
No,
true, I think, in a lot of biology. Like if you're trying to do computational biology, or something like that, you want to find a parameter value and you can't because it just doesn't exist. Someone did the experiment. You put
yeah,
it
they,
in.
they almost describe um, what you need, but it's missing. Like for my model, I needed it was attached by a point, for instance, and in the biology papers, I was getting attachment over an area. So you go throw your hands in the air and you go, I don't know. The data is not available, and it's just off by just. I think the biologists need to look at some of the engineering terms that engineers have used for centuries to describe things that are physical.
Yeah, yeah.
Just a comment.
Well, yeah, those measures already exist. But yeah, I think in biology, there, sometimes people aren't interested in those questions. Maybe it's a different class of questions they're interested in, like there's a convergence in like mechanical biology, which we saw in the Vertex papers. But a lot of times people are looking at different types of things, like gene expression, or they're looking at just like an organism and differentiation. So they're different questions. You notice in the differentiation paper, they didn't use physical terms as much. I wanted to kind of connect it more, but it was, you can imagine though that like in this final paper where you get these cell decisions being made, that this is all happening on an epithelial sheet or on some cellular sheet. They use the enteric nervous system as an example. So that's when you're developing this three-dimensional organ with multiple sheets, and you have these different cells that are differentiating on different ways. And so that's adding a lot more complexity there. Now, Dick made a comment that these look like differentiation trees, like in C especially, which is true. It's probably kind of where they might be going with that. But I don't think they make that connection explicitly. Also, I did look in a little bit into the C. elegans centitial topic that we talked about last week. And I did find some interesting things, although I'll have to put together a report maybe for next week. But yeah, I don't know how much data we would have for that, but I think there are some interesting directions. So, it might be that you have to put together a couple of data sets or get things from a couple of papers to get something together. And you may even have to do like some sort of pseudo data to see if that looks reasonable. So I'll do a report for next week. I'll try to do it for next week and see if I can, enlighten us a little bit more on that. Yeah.
I did a wide range of basement membrane attachments for my thesis, just to see what worked, because like I said, there wasn't any data, even though there was, but it was in the wrong, like I said, it was a dimension out. And I found that 10 newtons per meter was a bit soft for my construct, and 100,000 newtons per meter was a bit strong for spring attachments to the basement membrane. Anyway, that's just an observation.
Yeah.
But it was annoying, because there are measurements of the attachment to basement membranes, but it's not a point attachment, it's an area attachment.
Yeah.
Which is not possible in the program I was using.
Yeah, that is a good point, that, like, no pun intended, that, like, kind of think of a trijunction as a point, but it's really an area. There's up and down the axis, and it's not going to be like straight. It might be a straight axial line, but like it could bend, right? Like the edges of the cells could bend from curve, and that
Well,
would affect the
yeah,
structure.
that I didn't do scutoids or anything. I just did the simplest ones, like 3 different heights of cells, just straight up hexagons. But anyways,
Yeah.
oh, they're yeah, asking about
Oh yeah, the time.
daylight savings time.
Yeah, daylight. I don't know if anyone who joined at the top of the hour have anything they want to present or add to the meeting.
The thing is, I was checking whatever Mehul have done last week. Last year. So basically, he tried to like build some hypergraph and overall graph. So what is the next? Like I thought about if we can have a GNN or some GNN based model, and that will take those like graphs, whatever is already built, and process, and maybe because it is a temporal data, if we can add some kind of temporal graph model, maybe some discrete time temporal graph model like evolve GCN or something like that, or some updated model. Is it that, I mean, that area, is it okay to have, like, see, check, something like that?
Yeah, I think that, that's something we've been working on. Kind of, if you look at the other project from last year, the growing GNNs, that
Okay.
is going to have like some materials for like a temporal graph model, although it's not maybe not the best one. So I think let me go back to the repository here. So this is the growing GNN where we have this 3-dimensional space, and the animation isn't running, but it starts to grow from like 3 cells to 10 cells to 20, and you get these connections.
Okay.
And one of the advantages of having like an explicitly temporal graph is you could take out like each stage of the nomenclature and just present that, and that you would have like different slices, and so you could like
Yeah.
summarize the previous states and then have the current state as the graph, and then that would be a good temporal model because it would allow us to separate out the stages of each, what you would see like under a microscope, but also have that representation of the previous states, which is also
Okay.
useful.
Okay. So, is there any kind of prediction, you are trying to do, or just representing a graph is fine, or like what kind of direction you are thinking?
So I mean, we for now we've been visualizing things, and but eventually, of course, you want to predict things on graphs. You want to have things.
Yeah.
Well, in this case, maybe causality isn't like the thing, but maybe it is, because why are we building the graphs? Because we want to know sort of the interactions between cells. So we want to know that spatial relationship, but moreover, there are a lot of things that happen in space. So like the arrangement of cells in a sheet, which we were just talking about, or signaling, which is molecular signaling between cells, using those centroids as the representative points. So the farther away they are from each other, the weaker the signal, the closer they are, the stronger the signal. So there are a lot of reasons why we would want to model it with a graph, and then we can answer questions. So I mean, you could take like a graph and you could say, I want to build a model of interaction, with some signal molecules. So each vertex has a signal that leaves the vertex out into space and decays uniformly with space. So the farther away the other centroids are, the less effective they'll be, the less connected they'll be. And then, but you have that with the graph model, but you also have some sort of signal that transforms the cells in different ways. So you might have like a state change from red to blue. And you say, okay, if cells that are closer to this cell that's emitting the signal are redder than the other cells that farther away. You can see these patterns form. And that's kind of one of the things that we want to get out of these networks. It's these kind of relationships, but also these mechanisms for pattern formation and for other things.
Okay, yeah, understood. So yeah, I get the point here. So one thing I was thinking, like as with time it is changing, and the embedding, as you call, key, the uh, like cell change, the color will change. So based on some embedding change, the color will change. So what if we will predict the next state or something like that? I already know till 158th timestamp what is happening, and I am trying to predict after 158th or at 168th time what will happen. And there will be some kind of semi-supervised learning or similar kind of thing. And if we can predict using some discrete time temporal graph models, that can be one area I was thinking.
Yeah, I think there's literature on that, on on predictive temporal graphs, because you're using I guess maybe a Bayesian model or something. Basically you have the state that what the state is going to be going forward, or you could even build pseudo data where you build a lineage tree and you say these are where the cells divide, and then you sort of train the model on that, and you then try to predict what the next state should be given a signal. Given like 128 cells, what is the 152 cell state look like, which involves some asymmetric divisions, and the algorithm has to get to that, and it gives you these different outputs. You can say, oh, that looks plausible. We can do something like that. That would be good, because I think it's something that sounds kind of trivial when I don't say that these cells have a function and that eventually they need to be there for the adult to be functional. So yeah, that would be an interesting direction. There are a lot of things at the temporal organization of these graphs that kind of mimic what we call lineage tree, and we haven't really explored that too much.
Okay, if that can be a direction for this GSOC project.
Yeah.
So then.
Go ahead.
Okay. So then I can read the literature. I know some literature on that, and maybe I'll explore more and try to connect, and maybe I'll talk about it more in next meeting or so. Is it fine?
Yeah, that would be great. Yeah, I'd like to see the presentation on it. Yeah.
Okay. Yeah. Thanks.
Yeah.
Thanks, really.
Okay. So we had some comments in the chat here. Actually, it was a paper. I don't know what this paper is. Oh, this is a paper by Hirsch et al. On the archive, tracking biweekly supervised learning and graph optimization for whole embryo C. elegans lineages. This was Mehul posted this in the chat. So this is actually where they're training a model on all nuclei of an embryo using dense fluorescence microscopy, which is also noisy. So you have to get the nuclei segmented out of the image, and then use that as your training data. And that's what we're doing with DevoLearn. And then, so this is their method for doing this with weekly supervised learning. And they kind of talk about so I guess, Mehul, did you want to talk about this a little bit, or?
Yeah, sure. I can tell one or two lines. So what Joy was asking was he was telling that he wanted to apply temporal graphs over C elegans data. And you mentioned that since we have like discrete timestamps available from like 0 to 180 something, we can. So so this paper is doing something similar, but what it is exactly doing is just give me a second. I'm just like seeing the paper right now.
Okay.
It is doing tracking of cell lineages over the like development of C elegans. So I thought it is like quite similar to what Joy is like interested in knowing and doing.
Yeah, I think it is. So yeah, they're just trying to, because a lot of this segmentation is going to be prediction. So it's like what cell should be in the next frame. If a cell divides and you have noise in the image,
Yes, exactly.
then you have to know what a centroid is and what noise is, because you don't have the ground truth of this cell is actively dividing. Sometimes you take images that are on either side of that, and you can't capture like the dynamics directly in the microscopy images. So you have to predict all of that and what's most likely to be a centroid versus noise or something else. And then this is where we don't have the labels. So this is in C. elegans we have nomenclature labels. So that's a good thing, but the problem is that sometimes we don't have those in the microscopy images. We have to attach them later. So if we have a noisy image, what are we attaching? What label are we attaching to what? And that's where we can have problems. And the same thing holds true, I think, if you're simulating an embryo, if you're like simulating a bunch of centroids, is this like a plausible outcome?
Yeah, I get it. Okay.
Okay, get the challenges also. Okay.
Okay. So yeah, if people are interested in presenting on that next week, we can talk about that as well. I don't know, Lalith or Aniketh, do you have anything to add? Anything to update us on, or Divanshu as well?
Yeah. Hey, Bradley.
Yeah.
Yeah, sorry. I didn't get the thing about daylight savings time being active. Sorry, I got a bit late. Yes. So I was actually, I talked to Paki about something, and she came up with she suggested an idea to come up with a GUI for like visualizing how the graph evolves in real time. So I actually wanted to kind of explore that particular aspect of aspect, and maybe try to make it a project for GSOC this year. Would that be a good direction to do to take it up in?
Yeah, I think so. That would work. We did some explorations with the different topological data analysis, and we have that approach that we were taking. So this is back in the growing GNNs. Let's see if I can find it here. Actually, no, it's back here. I think it's in the stage 2 folder, the stage 2 directory. This is KMapper, where we're doing some topological data analysis, but this is just a fork of a previous project. Basically, what we want to do is create this visualization, which is where you have the cells by nomenclature, and then arranging it in different ways. So like part of a graph, or part of graph theory, part of network theory, is mapping things out in space. So there are different types of layouts. You have force layouts, you have circular layouts. Some are more sort of realistic with respect to the embryo. Some are easier to interpret with respect to looking at it by eye. So having a GUI there, which would allow us to build these different topological representations, would be good. Now, KMapper is there's like there's a repository here, and they have like a web interface, and I can't bring it up right now. But basically, the web interface is very limited in the different types of visualizations you can do. I think this Kepler mapper repository, it allows you to modify it if you get into the code, and we can host it on our website. But you can play around with the different types of visualization, and it involves topological data analysis, which is of course another step, and you can decide how far down that road you want to go. I would suggest probably just kind of a basic set of representations, but that's up to you. So I would like to build into what we have, but also I think it's a good idea to pursue, and I don't want to constrain you too much on how you do that, but I do want to have I do want to kind of do the things that we already have, and kind of expand those out. So
Yeah. Okay. Thank you.
all right. So if there are no other comments or questions, let's call it a meeting. Anyway, so I'll send out some materials, and if you have any questions, let me know, email or Slack, and see you next week. Thank you.
Thanks. Bye bye.
Thanks. Bye.